because I don't want you guys to wait too long. And if anyone just joined us after, they'll just look at where we stopped. Okay, so today I wanted to go over all of glycolysis and gluconeogenesis. We went over TCA last week mainly because glycolysis happens before TCA, but TCA tends to connect everything in the cell, which is why we go over it first and then we go over glycolysis. Okay, so this is everything that happens in glycolysis and everything we'll go over. Something I should mention, so here we have all the different substrates in pathway, and then we have these three main enzymes, which are irreversible enzymes in this pathway, which will play a big role. The other ones we'll also discuss, but those ones are mainly irreversible. Glycolysis is really ancient, and it's used by every cell in the body, which is it's mainly used to produce ATP, and it also produces electron carriers that can go in the electron transport chain and produce even more ATP. So if we run aerobically, so this is our product right here, pyruvate. So if we run aerobically, this molecule enters the mitochondria. So this is the mitochondria over here, and it goes through TCA. So this is like your pyruvate dehydrogenase complex and all the enzyme in TCA. So that's if you have oxygen. So if you have oxygen, we'll go into TCA. If we don't have oxygen, this pyruvate is converted to lactate. This mainly happens in muscles, which we'll go over all of those reactions. Another thing to point out is we start with a 6-carbon molecule and it splits into 2 pyruvates. So we get 2 3-carbon molecules. So that's kind of a general overview of everything we'll cover in this pathway. Okay, so to start glycolysis, we need glucose. So glucose is usually in the blood, and then it enters the cell. Different cells have different GLUT. They're called GLUT transporters, and that's how they bring glucose inside the cell. You guys don't really have to know much about GLUT transporters. Just know that different tissues in the body use different GLUT transporters, and they each have different affinities. So we have red blood cells here, and red blood cells, something important about them is they don't have mitochondria. So any glucose that enters the cell, it goes through glucose 6p, and then it goes to pyruvate, and then goes to lactate. So any glucose that enters red blood cells ends up functioning anaerobically because they don't, even though they carry a lot of oxygen in their hemoglobin, they don't have mitochondria to run electron transport chain or the citric acid cycle. So any glucose that enters red blood cells tends to be converted eventually to lactate. Okay, so now next we have the brain. So the brain is runs primarily on glucose. So most of the food we eat and the glucose we take into our body, I think like the number is like 25% of it ends up in the brain to run glycolysis. So your brain needs a lot of glycolysis, runs a lot of glycolysis, and requires a lot of glucose. So glucose enters through the GLUT transporter in the brain. It can be converted to glucose 6p, and that could also enter this is the pentose phosphate pathway, which is called PPP. And I also forgot to mention red blood cells can also run the pentose phosphate pathway. Glucose 6p is then converted to pyruvate, and then pyruvate is converted to acetyl-CoA, and would be your like TCA. And acetyl-CoA enters TCA, and then you get your electron carriers for the electron transport chain. Adipose tissue is really similar to the brain, except it can store glucose as glycogen. So after glucose is converted to glucose 6p, that can go and be stored as glycogen. And it also runs aerobically, converts to pyruvate and then acetyl-CoA. Liver is going to be your major regulatory organ. So your liver, one of its main functions is it regulates blood levels, the glucose blood levels. So if glucose levels in the blood are low, it will try to run Gluconeogenesis makes glucose and then releases to in the blood. If the blood has really high glucose levels, it will try to run glycolysis and use all that glucose. So your liver has a main regulatory function in regulating blood glucose level. Okay, so the reactions of glycolysis are mainly divided into two stages. So you have your priming stage and your payoff stage. So in the priming stage, which is right before we split, so up until here. So this is the priming stage. We invest 2 ATP and we create really high energy molecules that we can use to make ATP later on. After we split right here, this is done by aldolase, which we'll discuss later. We perform oxidation reduction reactions and we make 2 ATP in each direction we go. So we make 4 ATP total. So in net profit of ATP, we used 2 ATP and we made 4. We made 2 ATPs, and then we also have some electron carriers that we made. So the net overall energy that glycolysis contributes is 2 ATP totals. Okay, before we start on first reaction, do you guys have any questions on the overview of glycolysis? Okay, we'll continue. So the first enzyme is hexokinase. So what this enzyme does is it takes glucose, uses ATP, and it adds a phosphate on carbon number 6. So glucose, like I said, glucose enters through the GLUT transporters, and then we add this phosphate. So what this phosphate does is glucose here is a non-polar molecule. Once we add the phosphate, it becomes polar because the phosphate has the negative charge. This allows the glucose 6-phosphate to be trapped in the cell. So now we can't, if we just leave it as glucose, glucose can exit the transporter and enters, wait, someone's trying to join. Okay. So if glucose is by itself, it can actually leave and the cell through the transporters, because the transporters function via concentration gradients. So we add the phosphate to trap the glucose inside the cell. So hexokinase actually exists as 4 isoenzymes. So there's 1, 2, and 3, and 4. 1, 2, and 3, so 1, 2, and 3 have very low Km, so they have high binding affinity. The liver uses a different form of this enzyme, which is isoenzyme number 4, and it has its own special name. It's called glucokinase. So let's talk a little bit more about glucokinase. So it's a homodimer. That means it has 2 subunits that are the same. That's the 2 subunits is the dimer. Homo just means they're the same, and it binds its substrate via induced fit. So this enzyme has a higher Km, so it has a lower affinity. And I'll explain why this is important for your entire body and why it mainly exists in the liver. So when we convert glucose to glucose 6P, the other isoforms of hexokinase, this glucose 6P can actually go back and inhibit hexokinase, right? So in the liver, its main function is to regulate blood glucose levels, right? So let's say we have really high levels of blood glucose, and we want the liver to lower that. If the liver is running really fast glycolysis, it will make a lot of G6P, right? If the liver runs hexokinase, the isoforms 1, 2, and 3, this G6P will go back and inhibit the enzyme in the liver. So the liver has its own, the reason the liver has this enzyme is because it doesn't get inhibited by glucose 6-phosphate. Does that make sense? Okay. Another way that the liver can regulate glucose levels, so it's done transcriptionally by an enzyme, by a protein called glucokinase regulatory protein. GKRP, that's how it's abbreviated. So what this does is, so here we have the nucleus, and GKPR is over here, or RP is over here, and glucokinase is right there. Okay? If we have high blood glucose levels, GKPR acts on glucokinase and it sends it in the cytoplasm. That way it can start running glycolysis. And if we have low blood glucose levels, it sequesters it in the nucleus, because we don't want to run glycolysis if we have low blood glucose levels. And like I said, this enzyme is used by the liver and beta cells of the pancreas. Sorry. Do you guys have any questions on that? Okay. So here, I kind of just added this just to show you how glucokinase works. So if our blood is at 2 millimolar, the glucose concentration, and inside the cell, not inside the cell, this is the Km. So if we have low blood glucose, glucokinase is not going to be active. Not active, right? Because if we have low blood glucose levels, we don't want the liver to run glycolysis. On the other hand, if we have high blood glucose levels, glucokinase is going to be activated. That way, it can the liver is able to lower the blood glucose levels. So that's one way, one reason why the liver uses glucokinase instead of the hexokinase enzymes. And if it's in the middle, it runs like intermediately. It's not too active and it's not inhibited. Okay, so another way, so the liver has multiple ways where it regulates glucose levels. So this is our glucokinase enzyme. This is an enzyme in gluconeogenesis, so this is gluconeogenesis, which we'll discuss later. So what the liver can do is it can convert this glucose to glucose 6-phosphate, and it can, if we have high blood glucose levels, and we can also run the opposite direction if our blood glucose levels are low. And expression of those two enzymes, the glucose 6-phosphatase and the glucokinase, is altered by insulin. You guys will go over insulin and glucagon later in the semester. Just know that insulin and glucagon act on a lot of the enzymes we'll talk about today. Okay, so now we made our glucose 6-phosphate. Our next enzyme is called phosphoglucoisomerase. What it does is just converts this 6-carbon ring to a 5-carbon ring. So 5-carbon ring from a 6-carbon ring. This enzyme is reversible. It's an isomerase. So something special about this enzyme is it requires an OH at the carbon number 2, right here. And if this OH is not present, glycolysis will not run. This reaction won't happen. So a lot of times, what researchers do, or like treatments for cancer, is they add a fluorine instead of the OH at carbon 2. This fluorine stops the metabolism of glucose, and it also allows us to track where tumors or cancer cells are, because tumors and cancer cells tend to use a lot of glycolysis, and they run glycolysis really fast. So if we're able to stop their glycolysis metabolism and track where they are, this helps us serve as a really good treatment for cancer cells. And then we made this molecule called fructose 6-phosphate. Okay, so now after we made fructose 6-phosphate, we have another irreversible step, and it uses ATP. So this is the second ATP that we used. Hexokinase or glucokinase uses the first one. This is the second ATP used. And we attach the ATP at carbon number 1. So this enzyme is the committed step in glycolysis. So enzymes can be irreversible, but usually only one enzyme is a committed step in a pathway. So hexokinase that we talked about earlier is a irreversible step, but it's not a committed step, right? Because here, let's go back. This glucose 6-phosphate can go into other pathways, right? Once we go through hexokinase, our glucose 6-phosphate can end up in the pentose phosphate pathway. It can there's other pathways here that are not talked about, but G6P can enter other pathways. Once we make this fructose 1 bisphosphate, we're committed to running glycolysis. So that's the difference between a committed step and like an irreversible step. Committed step is once you go through it, you're committed to that pathway. And it also uses the divalent Mg cation. Okay, so this enzyme, since it's the committed step and it's an irreversible one, it's regulated heavily by a lot of stuff. Before I talk about that, if you guys have any questions, you can just interrupt me whenever you want. I'm not gonna get like upset or anything. Okay, so this is the enzyme here. It's a tetramer, which means it's made of 4 subunits. That's what that tetramer is. And it has 2 different states. So it has an R state and a T state. The R state is the high affinity state. So that means the enzyme is running fast when it's in the R state. And it also has the T state, which is the lower affinity state. If something's in the lower affinity state, it's running slower. So it's one thing it's regulated by is allosteric regulators. Before we talk about allosteric regulators, it's important to know that this enzyme has catalytic sites and allosteric sites. Catalytic sites is where our substrates bind. So these bind the catalytic sites. And then allosteric sites are the ones used to regulate the enzyme. Because we will see that ATP can actually bind to both sites, and it has a different function depending on where it binds. So negative allosteric regulators, they promote the T state, the lower affinity state, and those are citrate and ATP. So citrate, this is our, we go acetyl-CoA oxaloacetate, we make citrate in a citric acid cycle, right? So this is made in the mitochondria. And glycolysis happens in the cytoplasm. If citrate starts to accumulate so much that it starts leaving the mitochondria, that means we're running aerobic respiration really fast, and we need to slow it down. So citrate goes back and inhibits PFK. Same thing with ATP. If we have a lot of ATP, that means we're in a really high energy state, and we don't need to be running glycolysis right now. So ATP and citrate will bind in the allosteric sites and inhibit the enzyme. We also have positive allosteric regulators. So one of them is AMP. Think of AMP as opposite of ATP. I'll talk about the Pasteur effect later on, but if ATP is high, usually AMP is low. And if AMP is high, ATP is low. So AMP is kind of like the opposite of ATP for now. So if we have a lot of AMP, that means the cell is in a low energy state and we need to start running PFK. And then another one is fructose 6-bisphosphate, which I'll talk about later in more detail, but it promotes the high affinity state. And like I said, I put at the bottom here so no one gets confused. So ATP can bind allosterically and at the catalytic site because it's one of our substrates right here. So it can bind at the catalytic site and allosteric site. And all of this can also be regulated by insulin and glucagon. Okay, so this is the Pasteur effect. This allows glucose to be used by other parts of the cell. What I mean by this is if we have, if we're in a really high, the main point of glycolysis is to generate energy for the cell. If we have a lot of ATP and a lot of energy, the cell doesn't want to waste more energy into running glycolysis. So it has this system, it's called the Pasteur effect, which tells the cell if it's in a high energy or low energy state and what the cell should be running. So, I have a lot of ETC activity. So if we didn't go over ETC, but ETC produces ATP. That's all we need to know for now. So if there's a lot of ETC activity, a lot of ATP is produced, and this ends up slowing down PFK. And another name for ETC is oxidative phosphorylation. If you ever get confused, they pretty much refer to the same thing. So oxidative phosphorylation, electron transport chain, ETC, that's all the same thing. So this negatively regulates glycolysis, because if we have a lot of ATP, we don't want to be running more glycolysis. So this is the equilibrium that tells the cell if we have a high energy or low energy state. It's maintained by an enzyme called adenylate kinase. And what this enzyme does is it takes two ADP molecules and converts them into an ATP and an AMP. And this is the constant here. So we can see from the constant that if AMP goes down, in order to maintain the energy levels of the cell, ATP would have to increase, and vice versa. So it's a way for the cell to know where the energy levels at the current moment. So the sum of the nucleotides in this are always kept constant. Okay, so here I added a graph. So if we have no ATP, or if you have low ATP, that means AMP will be high and PFK will run very fast. So we can see that it has higher affinity. And when we add ATP, the graph shifts to the right. That means we have a lower affinity. And if we have a mixture of both, it's kind of in between. Okay, so like in summary, I just added this at the bottom. So when we have low oxygen levels, right, we won't be making a lot of ATP in the electron transport chain. So ATP production falls. So we have no oxidative phosphorylation. Our AMP to ATP ratio increases. So we'll have more AMP and less ATP. This will stimulate PFK. So PFK will try to run faster to make more energy for the cell. In contrast, if we have high oxygen levels, our electron transport chain will be running a lot faster, so we'll be making more ATP. We'll have oxidative phosphorylation, and the AMP to ATP ratio decreases. And this will inhibit PFK. I know it's a lot, but hopefully this helps you guys understand it better. Like when I was first learning this, breaking it down into especially this right here at the bottom, like the simple steps helps me understand the whole process of how the cell regulates energy levels and stuff. Okay, so this is our PFK enzyme. This is run in glycolysis. This is fructose 1,6-bisphosphate, which is in gluconeogenesis. So those enzymes work in opposite of each other. If one of them is regulated, the other one will be inhibited. So like I said, ATP inhibits. So inhibits. The red line is inhibits and the green line is activates. AMP will activate. And then fructose 6-bisphosphate will activate PFK. I'll talk about this in just a second, the fructose 6-bisphosphate. I have the ATP and AMP, act directly on PFK, and fructose 1,6-bisphosphate directly inhibits PFK. Okay, I think I have, so this is the fructose 6-bisphosphate. This is the regulation by pH. Okay, so another thing that can regulate PFK activity is pH. This mainly applies in muscle cells. Okay, so in the absence of oxygen, right, in muscle cells, pH drops, right, because you're running anaerobic respiration, so you're making lactate, and that will produce more protons, the H plus protons in the cell. So this tells the cell that the muscle is contracting really fast, and it's using anaerobic glycolysis. when the pH starts to drop, because that means we're making lactate, pH is dropping, the muscle is contracting, and it's a way that once the pH drops, it tells the cell that the muscle is contracting really fast. And glucose is being converted to lactate. So what this does is this drops the pH, and then this decrease in pH, so lower pH is more acidic, right? This protonates a histidine residue in active site of PFK, non-active, in the allosteric site of PFK, sorry. And this increases binding of ATP at the allosteric site. So when the pH drops, this protonates a histidine residue, and it allows it to better bind the phosphate groups of ATP at the allosteric sites. Once ATP binds, this inhibits. We talked about how ATP binds at the allosteric sites and it slows down PFK. So as soon as ATP binds, this inhibits PFK. So this decrease in pH eventually decreases the affinity, and it also decreases the affinity for the substrate fructose 6-phosphate. So in summary, when the pH drops, this tells the cell that you're contracting really fast. The muscle is contracting really fast, and it slows down PFK. So just so no one gets confused, this is in muscle cells. So when you have no oxygen in muscle cells, this will inhibit PFK. In other normal cells, because like I said, low oxygen here will eventually stimulate PFK. In other cells that mainly rely on the electron transport chain, the low oxygen levels indicates that ATP production falls, and that will stimulate PFK. Hopefully that's not too confusing. Just know that when the pH drops, mainly in muscle cells, this will inhibit PFK.
Question.
Yes.
So this is all in connection to the Pasteur effect?
Yeah, the Pasteur effect is mainly related with the ATP and AMP levels, but all of this stuff regulates PFK.
Okay, good.
Does that make sense?
Thank you. Yes.
Okay. The last thing that can regulate PFK is this enzyme called PFK2. I don't know if you guys talked about it, but we talked about it last year, so I thought I would mention it. So what we can do, so this is PFK1 right here. PFK1 takes fructose 6-phosphate and converts it into fructose 1,6-bisphosphate. So another enzyme called PFK2, it takes fructose 6-phosphate and it makes this fructose 2,6-bisphosphate. And if you have a lot of fructose 2,6-bisphosphate, this will go back and inhibit PFK1. This tells the cell that, hey, you have a lot of fructose 6-phosphate, that even some of it is being made into fructose 2,6-bisphosphate, and it eventually inhibits the enzyme, the committed step in glycolysis. So this enzyme mainly has a regulatory effect. It's not involved in any other pathways as far as I know, so it's mainly involved in regulation of glycolysis. Okay, we move on. Okay, so now we made our fructose 1,6-bisphosphate. This enzyme called aldolase. What it does, it breaks, so this is our 6-carbon, it breaks into 3 carbon molecules. So it takes our 6-carbon molecule that we made in the payoff stage, in the priming stage, and now we start the payoff stage, and we do this by splitting our 6-carbon into 2 3-carbon molecules. So this enzyme is actually freely reversible, and it's an enzyme that pretty much starts the payoff stage. A thing I will mention is GAB over here is what continues glycolysis. DAB can be involved in other things, like we'll see it will be involved in glycerol 3-phosphate shuttle. So some of this DAB can be converted to GAB, and that can continue glycolysis. But GAB is the substrate that continues glycolysis. So there's different isoforms of aldolase. So we have the A, B, and C. A mainly exists in the most tissues, and it uses fructose 1,6-bisphosphate as the substrate. B exists in the liver and the kidney and the small intestines. The brain has the isoform C, and it uses fructose 1,6 bisphosphate. Isoform B, the one in the liver, can use either fructose 1,6 bisphosphate or fructose 1-phosphate. So this comes from fructose. You guys don't have to know the fructose pathway. Just know that the isoform in B can use fructose, which eventually converted to fructose 1-phosphate, and that eventually can enter glycolysis. Yeah, and is made when you start off with fructose. Okay, so this is GAP. So this is what continues glycolysis. The dihydroxyacetone phosphate is DAP. And what we do is we split into these two molecules. This DAP can be converted into another GAP. So we'll finish this stage with two GAPs. And each of them enters its own direction, and they both continue glycolysis. Okay? So the enzyme that converts DAP into a GAP is the triose phosphate isomerase. It's an isomerization reaction, and it doesn't favor either side. It's mainly reversible, but when we want to continue glycolysis, this is mainly going to be converted back to GAP. So GAP continues glycolysis, and DAP is used in other things in the cell, like the glycerol 3-phosphate shuttle. Okay, so now we have GAP. So remember, now we have this. There's two of these because we had a 6-carbon molecule, and it split into 2 3-carbon molecules. So we have two of these. So anything we make now will be doubled. Does that make sense? Because we have two GAP molecules when we split. So this is done once GAP is made, it's converted into 1,3-BPG, 1,3-bisphosphoglycerate. This is done by the enzyme glycerol aldehyde 3-phosphate dehydrogenase. You can just call it GAPDH. It's easier to write. A main thing about this enzyme, so this enzyme is actually reversible, but this enzyme is a little complicated. So it's reversible, but it makes this very high energy product that can couple subsequent reactions. But I'll explain why it's reversible in the next slide. But just know GAPDH is generally reversible. This enzyme also uses NAD, and we mentioned that GAPDH is glycolysis is in the cytosol. So GAPDH is an enzyme in the cytosol. In order for glycolysis to run forward, we need different ways to make NAD plus in the cytosol. And the cell can use three different ways that you guys will learn about to regenerate NAD plus back in the cytosol. And we'll talk about those. I have them in the next few slides. So we make this high energy molecule, and we can start generating now our ATP. Because we used 2 ATP in the beginning, now we can start generating back ATP. Okay, so this is the mechanism of the enzyme, and it's a little complicated, but I just added it to explain why this enzyme is reversible, even though it creates a high energy product. So the GAPDH has an active site cysteine, and this active site cysteine right here is eventually converted into a hemiacetal intermediate. This intermediate is oxidized to a thioester bond. So we go from the hemiacetal intermediate to the thioester bond. The NAD is reduced, and we make our electron carrier NADH, and we also generate this thioester high energy bond. This is the same bond that pyruvate dehydrogenase complex uses. This bond eventually transfers the energy that it has to 1,3-BPG. And the enzyme, I'm not going to go into too much detail on the enzyme, but the enzyme just mainly continues in a circle. So once the energy is made in the thioester bond is converted into 1,3-BPG, and then the cycle starts over. This is the reason why this enzyme is reversible, even though we make a very high energy bond that is able to be coupled with the next subsequent reactions. Hopefully that makes sense. I also added that this cysteine here, this will be helpful once you guys learn the pentose phosphate pathway. So this cysteine here, basically anything that acts with the cysteine can mess up the GAPDH activity, because this cysteine is very important in the mechanism of GAPDH. So an example would be reactive oxygen species. Reactant oxygen species can directly act on the cysteine, and this can inhibit GAPDH activity. Okay, so this is mainly, I just mainly added this just to explain why this enzyme is reversible, even though we create a high energy bond. Okay, so I mentioned that the GAPDH needs NAD plus in the cytosol to run glycolysis. So the cell mainly has two main mechanisms. I'll mention a third one later on, but cells usually have two main mechanisms to generate that. So remember when we split, we made a GAP and a DAP molecule. So the GAP molecules continue in glycolysis. The DAP molecule can function in this thing called the glycerol 3-phosphate shuttle, where it's reduced to glycerol 3-phosphate, and in that process, NADH is oxidized back in NAD in the cytosol, and this NAD can be used for GAPDH to run forward. So we can use this DAP molecule, we can reduce it, and in that process we oxidize NADH back into NAD, and then the cell can use the NAD for GAPDH to run forward. This also goes backward, so glycerol 3-phosphate can go back to DAP, and then FAD, which is a different electron carrier, is reduced back into FADH2, and then those electrons enter the electron transport chain. This actually bypasses complex 1. For now, you guys don't have to know that. Just know that this shuttle actually bypasses complex 1. Okay, so the next shuttle we have, I forgot to mention, this shuttle is mainly used in the muscle and the brain. So different organs use different shuttles. So this one is used mainly in the muscle and the brain. The next shuttle is the malate aspartate shuttle. This is used in the heart and the liver. This one's a little bit more complicated, but it accomplished the same thing. It generates NAD in the cytosol for GAPDH to run. So I'm going to zoom in real quick here. So here we have the matrix. Malate goes to oxaloacetate. So this is your TCA cycle, your normal TCA reaction done by malate dehydrogenase. Oxaloacetate can be converted into aspartate. This is a transaminase reaction. Aspartate can leave the matrix and enter the cytosol, where it's converted back into oxaloacetate by the same enzyme. So it's a transaminase enzyme. Then oxaloacetate is converted into malate in the opposite direction now. So the way this is done, it runs in this direction in TCA, in the matrix, but we want oxaloacetate to be converted back into malate in the cytosol. That way we regenerate NAD for GAPDH to run. Okay, so we go through this whole cycle just to make NAD in the cytosol for GAPDH to use. Okay, so most cells are actually, malate aspartate's shuttle is actually more efficient than the other one, so most cells use the malate aspartate shuttle. The reason some cells use the glycerol 3-phosphate shuttle is because it's less dependent on TCA. So in this shuttle, if there's if there's anything wrong with any of the TCA enzymes or something's inhibited, it can affect the malate aspartate shuttle. So some organs in different cells can use the glycerol 3-phosphate shuttle. That way they're less dependent on TCA. Does that make sense? Okay, we move on. So we made this 1,3 bisphospho glycerate. So this is our high energy. And now we're finally able to make our first ATP. So we use 2 ATPs in the priming stage. Now we can make our first ATP. This is done by the enzyme phosphoglycerate kinase. It takes 1,3-BPG, converts it into 3-PG. This enzyme is mainly is freely reversible actually, and it's coupled with GAPDH. It takes ADP, and then we take this phosphate and put it on ADP, and then we make ATP. So consumption of 1,3-BPG can actually pull forward GAPDH, helps it to run faster in the downward glycolysis direction, and it uses substrate level phosphorylation. So this is the first ATP made, but remember, because we had 2 of these molecules, right? We made 2 ATPs in this step because when we split, we had 2 3-carbon molecules. So now everything we make will be doubled. So this step actually gives us 2 ATP in return. Okay, so something I added here. I don't think we talked about it last year, but I'm not sure if Dr. Kanapka will talk about it this year. But I just mentioned it because I thought it was cool. So arsenate over here. This is the structure of it, and it looks like phosphate. And what this does is, let's go back to here, right? So GAPDH uses this phosphate, and it attaches it to a GAP. So arsenate can actually act as a phosphate, and it can bind instead of the phosphate that GAPDH uses because it looks like it. This actually, you skip making 1,3-BP, which is the high energy, this one, which is the high energy molecule that we generate ATP from. So this, if arsenate is acts on GAPDH, it will inhibit ATP synthesis, but glycolysis will continue to run, right? So we won't make a high energy molecule to regenerate to make ATP, but glycolysis will continue to run, which is, it's kind of weird to think about because now the cell is just running glycolysis and it's not making any ATP. But that will that's basically the something the arsenate can do in the body. Okay. So next we have mutase enzyme. It's the phosphoglycerate mutase. We take 3-PG, 3-phosphoglycerate. We convert it into 2-PG. This enzyme is freely reversible. So I added not really the mechanism, but how this enzyme works. So we take the 3-phosphoglycerate, and we actually make an intermediate called the 2,3-bisphosphoglycerate in the active site. And then this 2,3-BPG transfers a phosphate to the enzyme. And then the enzyme is able to the enzyme is able to transfer that phosphate. So then we end up with 2-phosphoglycerate. So we transfer the 3-phosphate to the enzyme, and then we regenerate 2-phosphoglycerate. So this is important because this can actually act on hemoglobin and release oxygen. And I'll talk about in the next slide here I have why this might be important for red blood cells. But just know that you need enough 2,3-BPG in the active site for this reaction to run forward. So this is important because we learned this in Biochem 1. So 2,3-BPG can actually decrease the affinity of hemoglobin for oxygen. So when 2,3-BPG is present, hemoglobin will release its oxygen. And 2,3-BPG is needed for glycolysis because it's needed in the active site for this enzyme to work forward. So if the red blood cells can actually couple this reaction in glycolysis and affinity of hemoglobin to oxygen. And then red blood cells can also use this enzyme. It's called 2,3-BPG mutase, and it can convert a different molecule called 1,3-BPG back into 2,3-BPG. So this is mainly important for red blood cells because 2,3-BPG can act directly on hemoglobin. Okay, so now we have our 2-BG. So this is already a high energy molecule, but what we do is we remove a water and we add a double bond, and we make an even more energy, a higher energy molecule. So this, like I said, this molecule is already high energy. It has a delta G of negative 17.9. But when we remove the water and add the double bond over here, we end up with a molecule that has a delta G of negative 61.9 when it's hydrolyzed. So this enzyme is free reversible, but we mainly just do it to make a higher energy molecule. So this is your phosphoenol pyruvate, or you can just call it PEP for now. So now we made PEP, so now we're on the last step. This is done by pyruvate kinase. So this is our third irreversible step. We had hexokinase or glucokinase as the first irreversible step. And then we had PFK, phosphofructokinase, as the second irreversible step. And the third irreversible step is pyruvate kinase. What it does is it takes PEP with ADP, and it transfers this phosphate to ADP, and we generate ATP and pyruvate. So this is the final pyruvate is the final product of glycolysis, and this can enter in many different things. It can enter the mitochondria and go into the pyruvate dehydrogenase complex, or it can be converted into lactate, and it can even be converted to alanine. So this enzyme is irreversible, so it's one of the enzymes that is regulated. So if it can be regulated by ATP, so ATP can go back and inhibit this enzyme the same way it did on, because we mentioned ATP inhibits phosphofructokinase, so ATP can also act on pyruvate kinase, and it's also regulated by post-translational modification. With like any phosphorylation on the enzyme will also inhibit it. It's activated by fructose 1,6 bisphosphate. So this is the product of PFK. So what this is called is feed-forward activation. If we're making a lot of fructose 1,6 bisphosphate, that means we originally had a lot of glucose, and a lot of glucose is being committed into running glycolysis. So when a lot of fructose 1,6 bisphosphate is produced, it goes back and activates pyruvate kinase, and it's called feed-forward activation. Okay, so this enzyme has two isoforms, PKM1 and PKM2. PKM2 is generally slower than PKM1. This is not really important, but I just thought I mentioned it. I mention it because it's related to pyruvate kinase, but I don't think you guys would have to know this. So the two isoforms are PKM1 and PKM2. PKM2 is slower. This is generally expressed when glycolysis is running too fast. So this is another way for the cell to regulate, I guess, glycolysis and enzymes in general. When if glycolysis is running too fast and it wants to regulate that, it transcribes and it expresses PKM1. And actually, cancer cells, because they run a lot of glycolysis, they express PKM2. So it's another thing related to cancer cells, and it's a way for treatments, because we know that cancer cells express this enzyme more than normal cells. It's a way for us to make a treatment against cancer cells. Okay, so now we made pyruvate. Actually, do you guys have any questions? Because kind of went through that a little too fast regarding any of the previous enzymes or anything. Okay, so we made pyruvate. Pyruvate can enter the pyruvate dehydrogenase complex, and it can be made into acetyl-CoA. So this would be your when if you want to run TCA. It can also be converted to, I don't think they have it here. Oh wait, they do. It's right here. Can be converted to lactate. So this would be your anaerobic. And it can also be converted to alanine. And alanine will be important if you guys get protein metabolism. And not protein metabolism, amino acid metabolism and amino acid catabolism. So pyruvate can actually be converted into alanine. And then it can also be converted to oxaloacetate. We talked about this enzyme, pyruvate carboxylase. So pyruvate carboxylase takes pyruvate and converts it to oxaloacetate. This is also a really important enzyme for gluconeogenesis, which we'll talk about once we finish this. Okay. So lactate dehydrogenase is the enzyme that takes pyruvate and converts it to lactate. This uses NADH, and it generates NAD. NADH is oxidized to NAD+, and pyruvate is reduced to lactate. So this enzyme is reversible. It's mainly used under anaerobic conditions. So if you don't have a lot of oxygen, it's a way for the cell to not waste the carbons made from glycolysis. And it also generates NAD. And this is actually the third way that the cell can regenerate NAD for GAP-DHTUs. So the cell runs anaerobically to not waste the carbons it's made in pyruvate. And it also helps regenerate NAD for glycolysis to run faster. This enzyme will mainly be discussed in muscle cells because muscle cells use this enzyme the most when they're contracting really fast. And then cancer cells also need to regenerate a lot of NAD to run glycolysis even faster. So cancer cells express lactate dehydrogenase more than normal. Okay. So this is kind of how I think you guys had a question on this in the homework as well. So this is the muscle cell. So we go through glycolysis over here. And then pyruvate can be converted into lactate by lactate dehydrogenase. So another way for the cell, so it makes lactate. We also regenerate NAD. The cell doesn't want to waste this lactate. And what it does is it sends this lactate in the blood. The lactate travels to the liver, and then we run gluconeogenesis. So we transfer lactate back into the liver. The liver runs gluconeogenesis, regenerates glucose, and then this glucose can then go back in the muscle cell and be used in even more glycolysis. So this is called the Cori cycle, and it basically just functions. It's a really good example of how regulation between organ systems work, because lactate is produced by the muscle, it travels to the liver, converted back to glucose, and then that goes back to the muscle again. So it's a really good example of regulation of organ systems. Okay, so I think this is from your guys' book, but I included it just to briefly mention that other sugars can enter glycolysis from different pathways. So over here, we have glucose, so this can enter via hexokinase. So this is everything we've talked about. Galactose can enter. You guys don't have to know this. Just be aware that other sugars can enter glycolysis through different intermediates. So galactose can enter. It's converted to UDP galactose and then UDP glucose and then goes to glucose 1-phosphate. Mannose can enter to fructose 6-phosphate, and then fructose can also enter to fructose 6-phosphate. I think you guys should just be aware that different sugars can enter glycolysis through different intermediates. Okay. So that was pretty much all of glycolysis. Hopefully I didn't go through it too fast. Now we'll go through gluconeogenesis because it's pretty much the same thing, except we just have to bypass a few reactions. So in glycolysis, we use 2 ATP. We regenerated 4 ATP, so we made 2 ATP total. Okay, so to go back from pyruvate to glucose, we actually need the cell wastes 6 ATP when it does this. So most of the enzymes, remember we said in glycolysis all of the enzymes are reversible except for 3, the hexokinase, PFK, and pyruvate kinase. So gluconeogenesis runs all the enzymes the same except for these 3 boxes over here. So those are the irreversible ones in glycolysis, and gluconeogenesis is just basically a way to go around this 3 enzymes. Okay. And I think I mentioned, so we said glycolysis happens in the cytosol. Gluconeogenesis happens in the cytosol and the mitochondria. And this is everything we went over in glycolysis. Okay, so gluconeogenesis. So it's mainly used by the liver and the kidney. Gluconeogenesis takes pyruvate or other things I'll mention and turns it into glucose. Mainly the liver wants to run gluconeogenesis to generate more glucose if we have low blood glucose levels. So if we have low blood glucose levels, the liver will try to increase our blood glucose levels. So it will run gluconeogenesis and it will inhibit glycolysis to help regulate blood glucose levels. So after a meal, you have higher blood glucose levels, then most of your tissues and liver in your body will be running glycolysis. When you haven't eaten in a while or you're starving, you have very low blood glucose levels. So your liver will be trying to run gluconeogenesis to help raise the blood glucose levels. And then we're not talking about glycogen storage for now, but just note that muscle and brain, they can run either glycolysis or they can store this glucose in as glycogen. Okay, so we can run gluconeogenesis via different things. So the first thing we can run is we can convert alanine to pyruvate, and then pyruvate can go back to glucose. Another thing we can start, so these are all precursors for gluconeogenesis. Another thing we can use is lactate. So lactate can be converted into pyruvate via lactate dehydrogenase, and that can be used for gluconeogenesis. And then another one I added is glycerol can actually be converted into glycerol phosphate, and then this can be converted into dihydroxyacetone. So this is DAB that is formed when aldolase splits. So remember, aldolase splits into DAP and GAP. Aldolase is the enzyme, and it helps split 1,6-bisphosphate into DAP and GAP. And then DAP can actually continue and regenerate glucose. Okay, so these are different precursors that we can use to start gluconeogenesis. Okay, so we can't run glycolysis and gluconeogenesis at the same time. Otherwise, this would waste a lot of energy for the cell. So glucose to glucose 6-phosphate, this is done by hexokinase. So this is the first enzyme in glycolysis. So this is done by hexokinase or glucokinase if you're in the liver. Glucose 6-phosphate goes back to glucose. It's an enzyme called glucose 6-phosphatase. And those enzymes, this is mainly expressed in the liver. Those enzymes cannot be running at the same time. Otherwise, you will have what's called a futile cycle. It just means you're running in circles and you're just wasting ATP and energy. Fructose 6-phosphate can go to fructose 1,6-bisphosphate, so this is PFK. And then this is fructose 1,6-bisphosphatase. And then PEP can be converted into pyruvate, so this is your pyruvate kinase. And then to go back to here, we use two different enzymes. So pyruvate to oxaloacetate, so this is pyruvate carboxylase. And then oxaloacetate to PEP, this is done by a different enzyme that we didn't talk about. It's called PEPCK. Phosphoenol pyruvate carboxykinase. You can just call it PEPCK. Kind of sounds like Pepsi when you say it. That's how I remembered it. Okay, so the three enzymes we need to bypass are the hexokinase enzyme, the PFK, and the pyruvate kinase. All the other enzymes run in the opposite direction. We need to somehow bypass these three enzymes to regenerate glucose back for the cell. Okay, so the first reaction we need to bypass is pyruvate kinase, and this is done by two different steps. So we take pyruvate, converts it into oxaloacetate. So if you see OAA, this just means it stands for oxaloacetate. It's easier to spell it. So it's done in two steps. So we take pyruvate, convert it to oxaloacetate, and this is done by pyruvate carboxylase. Oxaloacetate is then converted into PEP, and this is done by an enzyme called PEPCK. So we're doing this to bypass phosphoenol pyruvate being converted to pyruvate via pyruvate kinase. So we have to go through two different steps to go back to PEP from pyruvate. And we use oxaloacetate as like an intermediate in between. Okay, so I talked about this last time, but I'll go over it briefly. So basically, just how pyruvate carboxylate works. Basically, we have, so this is an arginine residue in the enzyme, and we have an aspartate, or not an aspartate, a glutamate, sorry. This glutamate deprotonates bicarbonate, so this is bicarbonate. Bicarbonate then attacks ATP, and we have this carboxy phosphate intermediate. The carboxyl group is then transferred to biotin, so this is biotin. And we have this carboxy group on biotin now. And then this, the carboxy group is transferred to phosphate, and you make PEP over here. Oh, I'm sorry, I ran this the other way. So after we have the carboxy phosphate over here, biotin grabs this carboxy group from phosphate, and we generate this phosphate and carboxy biotin. Then pyruvate down here grabs the CO2 that, from biotin. So this is biotin, and it has the carboxy group over here. So pyruvate comes grab the CO2, and we make oxaloacetate. So we're just pretty much transferring this CO2 group. It transferred to a phosphate, then it transferred to a biotin, and then finally it transferred to pyruvate, and we made oxaloacetate. Okay, and I wrote down the steps here, if it's easier for you to understand it that way. Okay, so now we have our oxaloacetate over here. We use an enzyme called phosphoenol pyruvate carboxykinase, or PEPCK. This enzyme takes oxaloacetate and converts it into PEP or phosphoenolpyruvate. This gets rid of the CO2 that's attached to oxaloacetate, and then we instead attach a phosphate from GTP, and we make the PEP over here. This reaction can actually take place in the mitochondria or the cytosol, depending on the substrate you start with. So this is a little complicated, and I have a whole slide on that. But just know that oxaloacetate can go back to PEP either in the mitochondria or in the cytosol, and that depends on what you start with. So remember how we talked about we need to regenerate NAD for glycolysis to run? Well, to run gluconeogenesis, we need the opposite. We need NADH, right? So we need NADH in the cytosol for glycolysis to run forward. And it's mainly done by how PepCK functions. So here we have pyruvate going oxaloacetate. So this is your pyruvate carboxylase. And then oxaloacetate can go to malate. So this is malate dehydrogenase. And then malate can be exported. And then it can be converted to oxaloacetate in the cytosol. And then this can be converted into PEP. So we start gluconeogenesis in the cytosol, and we made NADH in the cytosol for GAPDH to run backwards. So this is if we export malate. Whenever we export malate, we're going to produce NADH in the cytosol. However, oxaloacetate can be converted, I'm trying to find room here. So oxaloacetate can be converted into PEP in the mitochondria, and then PEP then leaves. So depending on what you start with will help tell you whether you're running PEP, whether you're running PepCK in the mitochondria or in the cytoplasm. So if we export malate, that means we need NADH in the cytosol, and we'll run PepCK in the cytosol. When PEP takes place in the mitochondria, this happens if you generally start with lactate. So let me draw this one out. So if we start with lactate, we convert pyruvate. We also make NADH. And then pyruvate enters the mitochondria, and then it can be converted to oxaloacetate. If we start with lactate, we already made our NADH in the cytosol, right? Lactate go into pyruvate, makes NADH. And then pyruvate enters the mitochondria, and then we can run PepCK inside the mitochondria. If we don't start with lactate, we won't have this NADH. So we need a different way to regenerate NADH for GAPDH to run backwards. So usually, if we don't start with lactate, we won't have NADH. And then that's when your malate is exported out, and then it's converted to oxaloacetate in the cytosol. You make NADH, and then that can go to PEP, and then this can start gluconeogenesis. So it depends on what substrate you start with. If you have lactate and it's making pyruvate in the cytosol, you'll have enough NADH and you can run PepCK in the mitochondria. But if you don't, you'll have to export malate to make the NADH in the cytosol for GAPDH to run backwards. Okay, hopefully that wasn't too complicated. Okay, so the next enzyme we need to bypass is PFK, phosphofructokinase. PFK takes the fructose 6-phosphate and converts into fructose 1,6-bisphosphate. So to bypass this enzyme, this is, like I said, gluconeogenesis mainly happens in the liver. So this enzyme is mainly expressed in the liver. And what it does is it takes fructose 1,6-bisphosphate, converts it back into fructose 6-phosphate. It uses water and it regenerates the phosphate. This enzyme is also regulated, and it's activated and inhibited by the opposite stuff of what acted on PFK. So for example, ATP inhibits PFK, but it activates this enzyme. Same thing with AMP. So whatever effect it had on PFK, it will have the opposite effect on fructose 1,6-bisphosphate. And all other reactions between PEP between PEP to fructose 1,6-bisphosphate, they're all run the same because they're all reversible, so they're just run backwards. The one thing I talked about is we have to make sure that GAPDH needs any DH in the cytosol to be able to run backwards. Okay. So this is the last reaction we need to bypass, which goes from glucose to glucose 6-phosphate. This is done by the enzyme hexokinase. And we talked about this is the first enzyme in glycolysis. The liver uses glucokinase instead. So this, when we are going backwards, so we got to this point. So this is all of gluconeogenesis. We're going upward. We generated G6P. G6P can be used in different other pathways. So it can, for example, be used in the pentose phosphate pathway, or it can continue in the last step and be converted into glucose to complete gluconeogenesis. So the liver would probably want to run in this direction to regenerate back gluconeogenesis. But just note that a lot of pathways stop at G6P because G6P can be converted into other things in the cell. Mainly it's the starting point for the pentose phosphate pathway. Okay? So this is done by the enzyme glucose 6-phosphatase. So this is the enzyme that takes the G6P and converts it into glucose, or the last step. So this enzyme is actually in the membrane of the ER. So what happens is glucose here, so this is the cytosol, this is where G6P is. G6P enters through this transporter. It's called, I think it's just called the T1 on this diagram. But it enters through this transporter, the T1 transporter, and it's converted inside the ER lumen back to glucose, and we also regenerate a phosphate. And then glucose ends up going back in the cytoplasm. So this is done, so the cell doesn't, it's just a way for the cell to regulate gluconeogenesis and glycolysis to prevent a futile cycle. Because if those two enzymes are both run in the cytoplasm, there's no clear way to prevent them running in a circle. So glucose 6-phosphate is actually in the ER lumen. So glucose enters, glucose 6-phosphate enters, converted to glucose, and then it exits in the cytoplasm. And then once we made glucose, now the liver can actually release this free glucose into the blood, and it's able to raise blood glucose levels. So gluconeogenesis is done when you have low blood glucose levels. And then, like I said, glycolysis is done when you have high blood glucose levels, so you're trying to lower it. So you're taking glucose in the cell and you're running glycolysis. Okay, and then we also have to transport this phosphate back to make sure our phosphate levels is as well in the cytoplasm are maintained at equilibrium. Okay, so that was the last enzyme we went through to get gluconeogenesis. This is the overall summary of gluconeogenesis. So to convert from pyruvate to oxaloacetate, we use two ATPs and GTPs, or GTPs depending on what the cell is using. And then from oxaloacetate PEP, that also uses two GTPs and two ATPs. That's the ones where I went through the mechanism for both of them. So both of them have used phosphate from either a GTP or ATP. And then this is the phosphoglycerate kinase. So it's one of the reversible reactions I talked about. It comes after GAPDH. So to run it backwards, we will require ATP for that. So it required us a total of 6 ATP to turn 2 pyruvates into 1 glucose molecules. And then just as a summary, glycolysis produces 2 ATP. And then this is the free energy comparisons between them. So you can see that it's negative 38 kilojoules for gluconeogenesis. So gluconeogenesis is the everything we went through. But if we were to run glycolysis completely reversibly and not use those bypass reactions we talked about, it would actually be non-spontaneous reactions. So all those bypass reactions we used helped us generate a spontaneous reaction to take place. And then glycolysis and gluconeogenesis are both heavily regulated. Because you don't want them to run at the same time, otherwise you'll just be wasting energy all the time. So this is all I have for you guys today. I can answer questions on the homework or any questions regarding anything I presented. So I'll be here for a little bit if you guys have any more questions on anything. Otherwise, you guys are good to go. No one has any questions? Okay, well, I guess you guys are just free to go then. I will upload this if you guys want to go back and watch it.
I have one question. Sorry, I was just like looking over.
No, you're good. You're good.
So for 17B, when it asks us to
Let me pull
explain how
I don't have it.
substrates would be labeled